Biology Part 169. The trophoblastic villi becomes surrounded by spaces called lacunae, full of maternal blood. An anticoagulant produced by the trophoblastic cells prevents the blood lacunae from clotting. 5 weeks after the implantation of the placenta is well established and the blood vessels grow out of middle mesoderm, layer of the embryo into the trophoblastic villi, forming chorionic villi where the exchange of material occurs. These vessels connect with the developing heart and circulatory system of the embryo by means of umbilical cord. At the end of 2 months, all the major tissue systems of the embryo are developed. At this stage, it is about 2.5 centimeter long and is called a fetus. During the gestation or pregnancy period of 40 weeks, the fetus derives all its food and oxygen from the maternal blood and discharge waste, carbon dioxide, in the urea back into it. The transport material is made easier by the partial breakdown of the trophoblastic cells of the chorion, which brings the fetal blood closer to the maternal blood and is also facilitated by the microvilli, which are found over the entire surface. There is no direct contact between the two blood systems. Oxygen and carbon dioxide diffuse between them according to naturally established concentration gradients. Oxygen moving into the fetal circulation and carbon dioxide outward from it. The transport of nutrients and waste material is controlled actively by the placental membranes and requires the expenditure of energy by the trophoblastic cells. While the placenta is permeable to small molecules, it normally acts as a barrier to proteins and other large molecules which might cross from the fetus into a mother's bloodstream. This is important because it prevents the mother's immune system from producing antibodies against the fetus in the same way that her immune system would reject any other foreign material. An exception to this general rule sometimes occurs in the case of blood group substance known as the rhesus Rh factor. As explained, if Rh antigens from the fetus cross into the blood of a rhesus negative, Rh negative mother, the anti-Rh antibodies will be made. Together with other maternal antibodies, anti-Rh antibodies will cross the placenta from mother to the fetus and may attack the baby's red blood cells, causing hemolytic disease of the newborn. On the other hand, the transfer of the antibodies is normally desirable because they confer passive immunity against a whole range of disease organisms to which mother has been exposed. In this way, the baby is protected from infection from the first few months of her life until its own immune system is properly developed. Although the placenta acts as a barrier to many potentially damaging substances, some harmful chemicals, and disease organisms to pass from mother to fetus.